Hello and welcome to our last session focus on cell signaling. As we have heard before, cells receive and recognize external stimuli and convert them into a response within the cell. And this is the process of signal transduction. The response is usually a change in one or more cellular activity. So if a cell receives a signal, this could lead to any biological function, for example, migration, cell division, or even cell death. And the series of steps that make up signal transduction are called a signal transduction pathway. What are the key concepts in signal transduction pathways? First of all, we need to assume that there are specific molecular recognition events, for example, between ligands and receptors or between adapters and transducers. Also, another key concept in signal transduction pathways are molecular switches, and the first example of such molecular switch are G proteins. Another molecular switch we are going to be looking at is phosphorylation of proteins by kinases. Last but not least, another molecular switch is dephosphorylation by phosphatases. This is removing of the phosphate group from a protein. So what are G protein coupled receptors? Again, those very specific receptors are molecular switches within signal transduction pathways. They are receptors with an extracellular domain which is able to bind a ligand, and this is what you can see here. Those proteins also do have an intracellular domain which couples to a G protein, which you can see here. After a ligand binds to this receptor, this leads to a dissociation of the complex, and the G protein is binding to another protein present in the membrane. This in turn activates the signal transduction pathway and produces so-called second messenger. So this signal received by the cell is transduced into second messengers at this stage. Finally, after a set of other intracellular events, this leads to a cellular response I mentioned before. Another molecular switch we need to look at is phosphorylation, because this is another very fundamental process common in all signal transduction pathways. In simple terms, phosphorylation is addition of a phosphate group to certain residues present at proteins. Typically, phosphorylation is linked to one of the three amino acids that have a hydroxyl group in the side chain, namely tyrosine, threonine, and serine. This is something you should try to remember. And the addition of the phosphate group is catalyzed by very specific enzymes called kinases. The removal of the phosphate group is catalyzed by another enzyme called phosphatase. Those are the key players in this molecular switch. Another key concept within cell signaling and signal transduction cascades is that between binding of the ligand and the biological response, there is usually signal amplification between reception and response. So, what are the key steps of signal transduction cascades? In simple terms, there are three important steps that you need to remember. And the first one is reception of the signal. Second step is the transduction of the signal conducted by relay molecules and second messengers. And the final step is biological response. So what happens in phase number one? During this phase, a biochemical signal or ligand is detected when it binds to a cellular protein or receptor at the cell surface of the cell. And ligands can be very, very different. They can be proteins, peptides, lipids, or small molecules. So this is a very, very generic term. Basically, any substance that is able to bind to a receptor on the surface of the cell, inducing thereby signal transduction cascades. Receptors are specific cellular proteins, and they are usually membrane spanning at the cell surface, and they bind the ligands. There is actually one exception of the rules, because there are also receptors present in the cytoplasm of the cell, and those receptors bind, for example, steroids. But this is something Dr. Cottrell is going to be talking about in his biochemistry lecture series. For now, please remember that receptors are cell membrane spanning proteins. They usually have a bit outside of the cell, and this is the portion of the protein where the ligand is binding. They have element within the membrane, and they have also portions of the protein present inside the cell. So what are common families of cell surface receptors? First of all, they are ion channel linked receptors, and they are in charge of transporting ions across the membrane. Moreover, they are G-protein linked receptors. I introduced this type of receptors before because they are common molecular switches in signal transduction cascades. And we also have enzyme link receptors, often called protein kinases. And those receptors are quite interesting because they can sense a ligand, but they also have kinase activity. This means they can phosphorylate proteins or themselves once a ligand is sensed. Again, a ligand binding to a receptor is another molecular switch. So what you can see here is a receptor in its inactive form. We have two monomers of the receptor, and without presence of the ligand, they remain inactive and the switch is off. However, after a ligand binds to a receptor, this usually induces conformational change of the receptor, allowing thereby the receptor to dimerize to form a dimer. This in turn activates activity of kinases present on the receptor, and the switch is on. After the system is activated, this activates relay proteins, produces second messenger, and this leads to a ultimate cellular response. So, how does transduction looks like? Again, the transduction is a conversion of one type of signal into another. The first signal or the first messenger is the ligand. The second messenger can be, for example, calcium. And this is usually happening during the process of transduction. So again, we have activation of a receptor by a ligand. This is our first molecular switch. And this leads to activation of subsequent transduction switches. And usually the signal is here heavily amplified because binding of one ligand usually results in production of many molecules of the second messenger. This in turn produces biological response. As I mentioned before, ligand receptor interaction at the cell surface is usually converted into lots of activity, lots of second messengers inside of the cell. So we have binding of the ligand to a receptor, activation of the receptor, and production of a lot of second messenger. And this step is referred to as amplification. After amplification, the final step follows, which is the cellular response. For example, cell growth, proliferation, migration, change in gene expression, mobility, or changes in metabolic activity. So, via signal amplification, one ligand-receptor interaction can result in a very strong cellular response, which is actually needed because our cells need to react to changes in cellular environment fairly quickly. So how does amplification work? It can not only use second messengers, for example calcium, but it also often uses enzymes, kinases. Because of catalytic mode of action of enzymes, for each molecule of an enzyme that is activated, many molecules of the product will be produced. So we have a clear amplification of the response. Moreover, if several enzymes are linked in a series, then there will be amplification at each step. And this is quite evident on the right-hand side of the slide. One molecule of the ligand can produce millions of molecules or messenger that are in charge of producing the response. Amplification can also be achieved by opening of an ion channel. When the first signal molecule, ligand, binds to a receptor, this opens a channel. In turn, the ions are able to enter the cell, act as a second messenger, and produce a response. So again, we have one ligand, the first messenger, and the ions entering the cell are second messengers. So we have a clear and fast amplification. Another example of signal amplification is enzymatic generation of the second messenger. For example, cyclic AMP. And this is usually the case if the molecular switch in the signal transduction cascade is a G protein coupled receptor. So I showed you this slide already before, so I'm going to repeat myself only briefly. After binding of a ligand or a signal molecule to a receptor, this leads to dissociation of the G protein from the receptor. G protein binds to another molecule in the membrane, which is an enzyme, and this enzyme converts ATP to many molecules of cyclic AMP. And cyclic AMP is a second messenger in this system. This in turn leads to activation of several protein kinases, and another step of amplification is added into the cycle. As you can see here, we can have many kinases that are arranged in a sequential order. So we have signal amplification at each step, leading to a very strong cellular response. So what responses could we end up with? In simple terms, the response can be almost any imaginable cellular activity. So what are the target response proteins? Usually, the proteins that are targets of signaling pathway bring change in cell activity, contributing to the overall response. And those can be metabolic enzymes, changing the metabolism. We can have cytoskeletal proteins that play a role in migration, for example, but target proteins can also be gene regulatory proteins. And we will hear a lot about gene regulation in one of our upcoming genetics lectures. For here, it is important to remember that those proteins that regulate gene expression patterns are in charge of controlling activity of many different genes. So binding of a ligand to a receptor can change the gene expression pattern in a cell quite heavily. And this is important in processes such as maturation of immune cells or differentiation of stem cells. Stem cells per se are quite primitive cells. They don't have a defined function in the cell. However, if they sense presence of certain ligands, they can change massively and differentiate. They can become a terminally differentiated cell that is now able to fulfill a function in the organism. And some of those proteins called transcription factors have over 800 target genes. Moreover, transcription factors can also regulate proteins that intensify the same signaling cascade. We are referring to this as a feed-forward loop. So, for example, activation of the transcription factor may increase secretion of the ligand, and we have the amplification of the signaling pathway here as well. In contrast, transcription factors can also regulate expression of proteins that inhibit the same signal transduction cascade, and we are calling this a feedback loop. And this is quite important because in the body, in the complex system, processes like proliferation need to be tightly regulated. And if this goes wrong, we can end up with disorders and conditions like cancers. So you don't need to memorize this overly complicated signal transduction pathway, but this is just an example illustrating the key signaling concepts. So what you're looking at here is a very, very important signal transduction cascade regulating proliferation of the cells, cell division. So we have the ligands that bind to monomeric receptor. This activates dimerization of those receptors, activity of kinases, phosphorylation. This in turn regulates a lot of downstream kinases, so we have amplification at each of those steps. This regulates activity of transcription factors that are in charge of producing gene products that regulate proliferation. With this, I would like to end up this session, and I'll see you in our lectures dedicated to genetic content very soon. Thank you very much for your attention.